The overall goal of these procedures is to examine the physiology of worker honeybees in association with the worker's role and the status of their endocrine system.
This method, which has been widely used for the analysis of worker behavior, can also help answer key questions about the relationship between physiology and the role of worker honeybees. The main advantage of this technique is that it is relatively easy to perform.
To create 2 single-cohort colonies, begin with 3 standard colonies and collect newly emerged worker bees from each of the 3. Using tweezers, regularly inspect the colonies for the appearance of pupae in the capped peripheral cells with brown eyes and pigmented cuticles. Once found, most of the pupae will emerge in the next few days. Then, brush the adults off the combs and remove the combs from the colony. Combine 1 such comb from each of the 3 colonies into an empty nuclear hive box. Establish 2 such hive boxes and incubate them at 33 degrees Celsius with a relative humidity of at least 60%. Over the next 3 days, collect about 3,000 newly emerged workers from each new hive box. To approximate the number of bees in each collection, weigh 5 randomly selected newly emerged workers and use their average mass compared to the mass of the collected workers to estimate the number of bees. Prepare new hive boxes with a comb that is loaded with honey and pollen from an unmanipulated colony and another comb that is empty. Next, label the thoraces of approximately 900 workers in each new colony using water-based poster paint markers. Then, transfer the collection of worker bees into these new hive boxes and add 1 queen.